Hello, everyone. My name is Temitope Joshua, and I'm a PhD student in experimental surgery. It's my pleasure to introduce our student speaker for today, Dr. Anthony Bismarck, who will be speaking on evaluating the need for a structural colonoscopy curriculum, supervised by Dr. Sander Lieberman. So Dr. Anthony is a board-certified general surgeon and is a second-year colorectal surgery fellow here at McGill. He is currently pursuing a master's in surgical education. His research focuses on procedural training in surgery, particularly improving how colonoscopy is taught and assessed in general surgery residency program across Canada. So without further ado, please join me to welcome Dr. Anthony Bismarck.
Thank you. Thank you for the presentation, and thank you for having me today. So today I'll be talking about evaluating the need for a structured colonoscopy curriculum. I have no disclosures to share, except that as surgeons, we don't just cut, we also scope. And during our training, we're exposed to colonoscopy. So colonoscopy is part of our training. And by the end of our 5 years general surgery residency, we have to be able to scope our patients. So a bit of background, and the current model of learning colonoscopy is basically the apprenticeship learning. So residents learn opportunistically during their training. They don't have any clear benchmarks on their progression, and most general surgery residents learn colonoscopy by observing and occasional hands-on exposure. So there's a lot of variability across training sites and across Canada as well. We have a lot of residency programs, and each program has their own method of teaching residents. So either you go with a surgeon, and then they show you how they do their technique, and then you scope with them, and you learn by just seeing and learning step by step. There's a lot of limited feedback as well. So after each case, usually a good thing is to sit to see what you learned during the case and then how to progress. And there's a lack of assessment tools as well. So by the end of residency, we don't know if residents are actually ready to scope their patients. So most programs, as I said, they rely on the traditional apprenticeship model. There's no nationwide curriculum that's implemented, and there's no evaluation standards. Now we have, for surgeries, for example, we have what we call EPAs. So we have evaluation standards for everything else except for colonoscopy. And data shows that graduating residents report very low confidence by the end of their training. So why is this important? It's important because colonoscopy is essential for general surgery residents. It plays a crucial role in both diagnostic and therapeutic gastrointestinal care. As I said, many residents feel inadequately prepared to perform colonoscopy independently after their training. And when we have poor technique, it can lead to missed pathology and then increased complication. And so ultimately, what's important is patient safety, and that's what we're trying to make better during our study. So to understand the need of a structured colonoscopy curriculum, let's take a quick look at what the literature has already told us about it. Multiple studies echo the same concerns as residents. So there's a lack of preparedness, inconsistency, and even patient safety concern. For example, here Kim and Al found that many general surgery residents report insufficient exposure to colonoscopy that raise concerns about competency at graduation. Other studies showed that the trainees have longer procedure time, more complication, and increased patient discomfort. So it's not just about the comfort of the patient, it's also about the pathology that you find. Both Gardner and Francesca reported that general surgery residents frequently express concerns about their ability to perform endoscopic procedure independently after their graduation. And lastly, Delisle, a Canadian surgeon, found that program directors acknowledge that these training gaps are existent, and there's a struggle in implementing a structured curricula due to research limitation. And so what are the challenges in colonoscopy? So as we said, there's a lack of standardization. There's no nationally standardized curriculum. There's inconsistent exposure. We have assessment issues, so lack of clear consistency benchmarks, competency benchmarks to evaluate readiness, and the residency constraint. So our idea with Dr. Lieberman is to try and see if during this master's we can come up with a structured curriculum for teaching colonoscopy for general surgery residents, something that is competency-based, progressive, something that's targeted at teaching and has assessment tools, something that's adapted to resident level, so R1, year 1 to year 5, and something that includes simulation, supervised cases, and of course, a lot of feedback. So the plan is, first of all, to start with a survey. So we just want to see what the landscape is out there, do some analysis, and then see what the assessment, the needs are. And then we will draft the curriculum and pilot implement it to have actual data. So our first part is to try to do a national survey on general surgery residents. So it's a mixed method survey cross-sectional design that has 22 enclosed and 6 open-ended questions that targets all general surgery residents in Canada. So that's our population. And what we are asking is, first of all, is there a need for a structured standardized colonoscopy curriculum? So do they actually need it, or they're comfortable with their current methods of training? We want to see what's working. What do they have in their program across Canada? What's missing? And what do residents actually want to see in a future curriculum? And so the purpose obviously is to assess the landscape of the colonoscopy training across Canada to see if there's any gaps in training and competency development, and ultimately to evaluate the need for this curriculum. That's a bit of some questions that we have included in our survey. So for instance, what's your current experience and what challenges do you face? Let's say, oh, I don't have enough hands-on practice opportunities. I don't have enough feedback when I do my procedures. I have limited access to simulation labs. I have difficulty managing patient discomfort. So the goal is to gather all this information and ultimately establish a foundation for our future colonoscopy curriculum development. We want to develop evidence-based recommendation for a standardized curriculum and to see what are the gaps as well. So their next steps, once we have the results of this survey findings, we want to develop the core elements and do a pilot study by implementing our new curriculum at 1 or 2 Canadian sites. So McGill will be one of them. And then we want to focus on the feasibility, progression on benchmarks and assessment tools. And obviously we want to collect the feedback and revise accordingly. So ultimately, this is about trying to raise the bar nationally. We want to put a program that meets the needs of today's learners while delivering safe and high quality care for our patients as well. So imagine a Canadian general surgery finishing residency with confidence, competence, and credentials in colonoscopy. We want formal teaching, objective evaluation, longitudinal exposure, and hopefully that's something that will be supported by data and evidence. Thank you for listening.
Thank you for your presentation. So we have a question in the box and also, okay. So I'm just going to go with the question in the box first. And please, everybody, take a moment to complete the evaluation survey being shared on the screen. So Mashid asked, great presentation, Anthony. What is the main reason of lack of confidence of general surgery residents? Is it mainly due to the limitations in the training or maybe imposter syndrome?
So there's a lot of data about their confidence at the end of training, and the main issues is their exposure. So depending on the program, sometimes you have longitudinal exposure. You have 1 month where you put the resident and they scope every single day with a surgeon, and others where they just go 1 day in a week where there's a gap in the colonoscopy suite, and then they have their exposure this way. So there's no structure in the way they're taught how to scope. There's no benchmark. In the US, there's let's say they have numbers that they have to complete before graduating, but that's something we don't have here in Canada.
Okay, so the next question is by Jackson. You can please unmute to ask.
I think that was me. Great.
Yeah.
It was nice to see the research project presented. This kind of stems a little bit from the previous question. I wondered whether or not you're trying to collect in parallel with the survey data both people's perceptions of where they are in acquiring their skill and perhaps some kind of frequency of how often they have an opportunity to practice these skills. And the reason for trying to collect both of those answers is that, people have different learning curves as we've covered in the course. But the perceptions are important and relevant, but they're a subjective piece of data. And people can feel more or less prepared, partly as a result of what kinds of expectations they might have. There's a lot of other factors in there as well. So I think that getting at the subjective would be especially helpful if there's some more, it would still be self-reported, but if there was also some more objective and numerical, kind of answers or questions there. And I think getting at frequencies, for example, of opportunities to do procedures or the frequency with which they played a role in a procedure, et cetera, that might be a nice and complementary approach to take. The other piece is more of a comment, but I'm just going to go ahead with that anyway. And that would be where it sounds like it's a great project. You'll definitely get the survey out. And it sounds like getting a drafted curriculum together could maybe be possible. But I can't remember, Anthony, where you are in your master's. I wondered about the implementation. It sounds like that could be maybe a little bit ambiguous. So I wondered if you could remind us what the timeline is.
Yes. So thank you for your questions, Jason. And yes, so I haven't shown the whole survey, but the survey does include qualitative and quantitative data. So there's open-ended questions at the end for residents to report what's their current fears, what their exposure are, what do they want to see in an open-ended way of reporting it. And we'll have also the quantitative ones. And as for the timeline, I started my master's a bit later than everyone, so I didn't start it during the fall. I started during winter. So obviously I'm in my first semester, but we like the curriculum is already mostly drafted, but I think that survey will become very, very helpful in refining it and trying to implement it.
Wonderful. That sounds great, Anthony. And just a reminder again that qualitative and quantitative are on a bit of a different axis than objective.
Right.
But that's great and really exciting to hear about all the wonderful progress you're making. Congratulations.
Thank you, Jason.
Okay, so Anthony, you have another question by Fatima. She said she has, why did you choose a mixed method approach for this survey?
So a bit of what I said is we wanted to gather not only the data on how much they scope, what's their exposure, but also gather some idea and come up with common themes that they could have. Something we haven't thought about during our writing of the survey. And so that would allow us to refine our curriculum. So that's what it's a mixed method.
Okay. All right. So she has a follow-up question, and she's asking, how do you plan to integrate qualitative and quantitative data in your analysis?
So our analysis will be mostly descriptive. So we want to come up with percentages, means, etc. And then for the qualitative data, we'll gather all the information and we'll use coding to have common themes come up, and then do some analysis with the themes as well.
Okay, great presentation. So I have a question for you.
Yes.
So my question is, what do you see as the core competence that a structured curriculum should address in this case?
So the core competence would be, I think, patient-centered. So for me, it's when a patient is uncomfortable during a colonoscopy, it says a lot about the endoscopist. Sometimes, yes, the colon is difficult. Yes, there are curves. Yes, sometimes, depending on if you do it today or in 3 months, it's going to be completely different. But I think ultimately it's patient comfort. And so I think that would be a huge telling on how the endoscopist is scoping. So it would be, so other than the technical aspects, reducing the loops, trying to come up with water infused instead of pouring CO2 that inflates the bowel and provokes more pain. So all of this translates in patient comfort.
Thank you. Great presentation.
I do have a quick question, actually, Anthony. A very nice
Yeah.
presentation. And I can imagine as many of us get older, we probably want to know what's the outcome, and hopefully that could be improved, like you mentioned, in terms of the discomfort that patients receive, depending on the endoscopist. So I was just wondering, as more curiosity, are there differences in training modalities between institutes? So you mentioned you're going to do, your study design includes McGill and one other center, right? One other. So are there already existing differences in how how these people are trained?
Yeah, so now there's no fixed common method. So each center trains differently. Some of them gets trained by general surgeons, others get trained by gastroenterologists. Some of them have simulation labs where they can train on a fake colonoscopy model. So there's no proper way, and the training differs a lot. I trained my general surgery residency in Sherbrooke, and by the end of training, I was very comfortable in colonoscopy. And when I came here to my McGill and to colorectal fellowship, it's expected that we scope as well. And I have noticed that there's a huge difference in exposure and in scoping competency in residents, let's say here or in versus somewhere else. So the training is different depending on where you train. Yeah.
So it's literally the major differences are between the institutes, not within the institute. Like, I would imagine there are differences even within, or is just between.
Even within between residents, they don't have the same exposure. So so sometimes during your rotation, there's this amount of scope days, and so a resident gets lucky and then gets more exposed to colonoscopy. Others have more priority to go operate and train in the operating room. But ultimately, when you graduate, you have to be competent not only in doing a gallbladder surgery, but also in scoping.
Sounds good. Good luck.
Thank you so much.
Okay, thank you for today's presentation.
Thank you.
Our next speaker, I'm not sure she's here, Zara.
Yes. She's here. Yes, yes.
All right. So, okay, you may want to share your slide. Okay, so our next speaker is Zara Kadan, and today she will be speaking on structure pattern and transcriptional effects of integrated Epstein-Barr virus, reviewed by a long-read sequencing and RNA sequencing in nasopharyngeal carcinoma. She's supervised by Dr. Sapat Kumar Langatan, and Zara is a second-year master student in our division. So join me in welcoming Zara.
So hello everyone. Do you have my screen?
Not yet. It's the
Yeah.
Okay. So this is Zahra Cardon, and today I want to present my master's work focused on structural pattern and transcriptional effects of integrated episteme virus revealed by long-read sequencing and RNA sequencing in nasopharyngeal carcinoma. Nasopharyngeal carcinoma, or NPC, is a head and neck cancer subtype and is located in nasopharynx. Nasopharynx is an area behind the nose and above the throat, and its global distribution is highly uneven. It's considered a rare cancer across the globe, but in some specific area is considered prevalent, such as South Asia and North Africa. Its risk factors include genetic factors, viral infection, and environmental factor. Genetic factors like family history and genetic background, environmental factors such as dietary factors and consumption of salted fish or preserved food or exposure to pollution. Also, viral factor includes Epstein-Barr virus or EBV infection, which kind of concern almost 90% of the NPC patients. EBV can integrate into the genome and cause genome instability. It can also change host gene expression alternation. EBV is the strongest risk factor associated with NPC, and for understanding it is crucial to combat EBV-associated cancers. EBV is a double-stranded virus with a size of 172 kb. It has genes that are considered to be oncogenes such as EBNA1, LMP1, and RPMS1. EBNA1 is important for keeping the virus in latent stage. EBV is able to make a lifelong latency in epithelial and B cells. And EBNA1 can help the virus to reproduce. LMP is important for keeping the virus in late stage as well, and it's important as it can imitate some receptors and lead to cell proliferation of the infected cells. And RPMS1 is a non-coding RNA, and it can produce non-coding RNA and microRNA, which are important for immune invasion. But EBV tumorigenesis doesn't limit to these oncogenes, as it can integrate into the host genome and cause genome instability, potentially disrupting tumor suppressors and finally leading to cancer. So understanding how EBV can integrate into the human genome is really important to combat EBV-associated cancers such as NPC. For my master, we had 4 samples, 4 NPC positive samples, which consists of 3 cell lines and 1 tumor samples. For sequencing and finding the EBV integration, we use long-read sequencing to capture larger genomic area. The longer sequencing technology today can capture reads ranging from kilobase per to megabase per with 98% accuracy. So we opted for Oxford nanopore sequencing technique as it has a membrane with a lot of nanopore embedded in it, and there is a currency flowing through these pores. And then we have the single-stranded DNA is passing through the pore. Each base pair that passed through the pore, it changed the currency. And the device detects the currency changes, and at the end, based on these currency changes, we have our sequences. And this is steps called base folding that we determine which sequence corresponds to the currency changes. And after we have our sequences or our read, we move to next step. Our read is going to be in FASTQ file, and we want to align it to our reference genome with Minimax tool. This step is using our custom reference genome consisting of EBV and human genomes. And after the alignment, we do a quality control to see the alignment efficiency. At this step, we want to see whether the alignment happened perfectly. There is no mismatches or mapping to several locations, or big inserts can be identified and removed if they are dropping the quality. And after we are ensured of the quality of alignment, we move to finding a structural variants such as insertion, inversion, deletion, translocation, and duplication by swim. At this step, we can find EBV integration breakpoint into the genome. So when we do the alignment, after we want to see the presence of EBV in all the samples. And as we can see in figure A, we can see that EBV was present in all 4 samples we had. The whole length of the EBV genome was present. MPC 219 is our tumor sample, and MPC 43, C6661, and C17 are cell line samples. Then we wanted to see the integration breakpoint across EBV and human genome. In total, we could identify 36 integration breakpoints. And the integration breakpoint patterns seem random with no specific localization of EBV into the human genome. But we could identify integration hotspot in both EBV and human. For EBV, EBNA1 and RPMS1 are integration hotspots. And for human, we have chromosome 2 that has the most of integration happening in human genome. Then to identify the gene expression alternation around this integration breakpoint and to find fusion transcripts arise from these integrations, we wanted to do RNA-seq. For the first step and finding differential expressed genes, we had our raw data. Then we did a FASTQ quality control and then mapped it, did the count analysis, normalized, and found differential expressed genes. For finding the fusions, we use the aligned band file from previous steps. And after we have the aligned band file, we can find chimeric reads. Chimeric reads are the reads that one part align to one part of the genome and one part align to another part of the genome. And after determining these chimeric reads, we can do fusion calling using ARIBA. After we did RNA sequencing, we could find the gene expression alternation around integration breakpoints. So 44% of our genes around the integration breakpoints had gene expression alternations. These genes were involved in important pathways like immune regulation, cell cycle, and gap junction. Importantly, these genes expression alternation pattern had both upregulation and downregulation. Interestingly, we found some of these genes have upregulation, and they are important to oncogenes such as PIK3CA, CD96, and RH-GAP27. And we also have important tumor suppressor like HOT1 that is downregulated. So we saw both downregulation and upregulation of tumor suppressors and oncogene in their corresponding samples. And at the end, we wanted to see fusion transcripts. As I mentioned before, when we have the chimeric read, we can find the fusion transcript. We found both fusions in human and EBV human transcripts. For human, we wanted to find driver transcripts. And for finding driver transcript, we use Anofuse. Anofuse is a package that goes and look in all the human fusion and look for driver fusion based on the TCGA reports. And based on the known knowledge of oncogene or tumor suppressor. So if the fusion protein has a partner that is oncogene or tumor suppressor, it would be considered a driver fusion in anafuse. For EBV, we used Arigo to find chimeric that they are partly human, partly EBV, and we could find almost 496 fusion transcript across all 4 samples. And interestingly, we could find 2 fusion transcripts that correspond to our integration breakdown. So when we saw EBV integration, we found between 36 breakdowns that we had, we found 2 fusion transcripts arising from those integration that include the fusion of RAL-GAP A1 and LMP2A and link 00486 and RPMS1. RAL-GAP A1 is a ROGPTase that is important for cell proliferation and migration. Link 00486 is a non-coding RNA that can be important for cell transcription machinery. So these fusions can be oncogenic in NPC context. So I am open to any question we may have.
Thank you for the presentation. Question?
Yeah, that a very nice presentation. I'm curious about the fusion transcripts. So is that are unique to the EBV? So you found this is this like in all tumors that were infected with EBV latency, right, virus? They all contain the same fusion transcripts, or is this something that's random and how do you first answer those questions?
So across different samples, we have different EBV integration in different genes. So RAL-GAP A1, for example, happened in sample C17, and link 00486 integration happened in sample MPC290. So all the samples doesn't share the same integration breakpoint. So it's happening in different genes. And so they are from different samples. They are not sharing.
And is that related to the severity of the cancer?
For the cell lines, we don't have the severity in stage information, so I cannot say for sure that whether it's linked or not. But for the tumor, I know that the stage was 3, so it was pretty severe. But I don't know whether it's linked to the fusion transcript, if necessary.
And so these are novel? These are novel fusions that you have found. Okay, very interesting. And I would imagine, what's like the future prospects of this work? Do you plan on targeting these types of fusion? Is this one project to target them to see how that may affect the, say, tumor progression or growth?
Exactly. So the future direction can be that we imitate the fusion transcript and transfect the cells with it and monitor the growth and migration. Also, most of my project is prediction based. So for validating, we can use a lot of validation in the cell lines or in the mouse.
Exactly. Oh, good. Very interesting.
Okay.
Okay, thank you, Zara, representative. Is there any other question? No question. Okay. All right, thank you.
So Mike, can you start with the
Yes,
main
absolutely.
speaker, please?
Yes, it is my pleasure to introduce our guest speaker, Professor Coleman. So Professor Coleman received her a bachelor degree in mechanical engineering from the University of Rochester. She then received her master's degree in mechanical engineering and a PhD in bioengineering from Georgia Institute of Technology. She then moved onto the hospital for special surgery in New York City to complete her postdoc, where she studied methods to prevent mineralization of cartilage. Finally, she joined the Faculty of Biomedical Engineering at the University of Michigan in 2012 to form the Cartilage Healing and Regeneration Laboratory, where she combines the background in mechanics and tissue engineering to investigate methods to develop mechanically functional cartilage replacement and restore joint mechanics in physiologically normal conditions. So once again, thank you for accepting our invitation, Professor Coleman, and the floor is yours.
Thank you for having me, Dr. Grant. I appreciate your introduction. And today I'd like to talk about our efforts in our lab to improve stem cell-based cartilage tissue engineering. And we do that in a way that integrates synthetic biology and current tissue engineering applications. And I'm sure most of you know, sorry, there we go. What articular cartilage is. There we go. But for those of you who don't, it is the tissue at the ends of the long bones required for motion and distribution of loads throughout the joint. Typically, what we'll see is this beautiful white tissue that's shown on the bottom left with intense uniform staining, which shows the simplicity of this tissue of a single cell sitting in a very fairly homogeneous matrix. And so the efforts to engineer this were supposed to be easy, but has been fraught with difficulties that we didn't imagine. And one of them is to find a cell source for tissue engineering. And really the reason we need to do this is for addressing the burden of osteoarthritis. So as I said, we have beautiful white glistening tissue, but as osteoarthritis progresses, we see progressive joint-wide damage throughout the tissue, which eventually leads to a joint where we have damage distributed across the tissue, and the only solution for it is total replacement of the knee. So this is a worldwide disease that impacts 300 million people globally, and that was in 2017. So we know that the numbers are increasing significantly. And actually, it's expected to be the 5th largest cause of disability in the world by 2030. So it is a very significant disease that needs to be addressed. And of these osteoarthritic patients, we know that 12% of them were due to some sort of previous injury, such as an ACL tear, or as shown here, a cartilage defect. And that leads to something that we know as a post-traumatic osteoarthritis, or PTOA. And these defects aren't easily detectable because we can't sense them due to the aneural nature of articular cartilage, and so they're usually not discovered until some sort of orthoscopic procedure has happened for a different injury, at which point they've progressed to the point where they're starting to nucleate osteoarthritis. And so really our goal is to develop a clinically relevant adult MSC-based cartilage regenerative strategy to prevent the onset of osteoarthritis. I don't know why we have that. Unfortunately, there are 2 main challenges that I'll discuss in this presentation that have been preventing the clinical application of these cells. And the first one really has been the stability of an MSC-derived chondrocyte, which I've abbreviated as MDCH in this presentation. So as shown here in this paper by Rob Mock's group, when you take out articular chondrocytes shown on the top, you get robust matrix production over a long period of time. But with MSCs, you get very little matrix production, and actually what we see is a loss of matrix over time. And this leads to an increase in the mechanical properties of regenerative tissue generated with chondrocytes, but a loss in these mechanical properties when we create tissues with MSCs. And this phenotypic stability has been well documented and comes from the pathway that these cells have decided to go down. So typically what we'll see for articular cartilage is a chondrocyte, which is the main cell within the tissue. It's embedded in aggrecan and collagen type 2 predominantly, and these are the matrix markers that are very important for the mechanical function of the articular cartilage. So these are the ones that we are trying to build up as much as possible when we engineer these tissues. And when human MSCs are induced to chondrogenesis, they will initially make a tissue that's robustly expressing aggrecan and collagen type 2. But rather than remaining articular cartilage, they follow the pathway of growth cartilage. And growth cartilage is the cartilage, the precursor to endoconal ossification or long bone development. And during this process, the cells will proliferate, undergo hypertrophy, and hypertrophy is marked with this decrease in aggregan and collagen type 2, those required proteins for mechanical function, and an increase in metalloproteinases, which will also degrade the existing matrix. So we can't very well engineer a tissue with these cells as they are now. And so a great deal of work has been done in understanding how these cells differentiate through this process of a stem cell to a hypertrophic differentiation. And we know that this is mediated predominantly through members of the TGF-beta superfamily, as well as the Wnt family. And transcription factors that have been identified to be important in this process include SMAD3, SOX9, RUNX2, and these transcription factors are responsible for the transitions in the different cell states. If we look at a detailed analysis of these cells, we see that there are cell surface receptors and ECM components that identify each stage. And what we've noticed is that the mediator to drive hypertrophy of these cells is RUNX2. And the target genes, such as MMP9 and 13, as well as collagen type 10, are upregulated by RUNX2. And so we use these markers for hypertrophy. And we also know that RUNX2 is highly expressed during this process of endoconal ossification. And so really, we've been investigating a number of approaches to regulating RUNX2, including the influence of exogenous cues to regulate this process, as well as reprogramming MSCs to autonomously express gene circuit. And I'll focus on the second process during this presentation. So we could try, and we did try, and many of others have tried in the past to exogenously regulate RUNX2 transcription or activation through phosphorylation. And as we can see in this pared down schematic of RUNX2 regulation, there are quite a bit of physical cues and biological cues, cytokines, chemokines that will regulate this process. And so it's almost impossible to come up with correct combinatorial and temporally and spatially represented cues that can regulate RUNX2 in a manner that we want. So what we were looking for was something a little more, so we suggested or decided, well, the first obvious solution to this problem is just to silence RUNX2. And so we did using a short hairpin and RUNX2. And what we found is that when we silence RUNX2 in these cells, we decrease the amount of matrix that's produced, and we decrease the expression of those valuable structural molecules, collagen type 2 and aggrecan. And so we can't simply shut off RUNX2 because it's required for chondrogenesis. And secondly, the thing that we notice, and that's obvious in this bottom picture, is that there's a heterogeneous response in chondrogenesis of these cells, which means they're at different cell states, and we're treating them all the same way. So what I really wanted was a way that the cells could sense their environment and then modify their behavior based on that environment, essentially using the cell's own machinery. And that brought us to synthetic biology and tissue regeneration. So it was around this time that synthetic biology in mammalian cells was becoming coming out and into the literature. And we wanted to use this to regulate RUNX2 activity. It was also around this time that several gene circuits were created to reduce the effects of of inflammatory factors on matrix loss during cartilage tissue engineering. And so taking a page out of all of this research, we wanted to come up with our own genetic reprogram of chondrocyte phenotype by developing our own gene circuit. And essentially what we wanted was a cell autonomous way to regulate MDCH hypertrophy. And the way that we decided to do that was to create a synthetic RUNX2 negative feedback loop. And so just to give a brief overview of how that would work, we would hijack the natural process of RUNX2 transcription and translation within the body. So we know that when maturation signals are received by chondrocytes, we'll have transcription of RUNX2 and its subsequent translation, but we also have activation of RUNX2 through phosphorylation, which allows it to translocate into the nucleus and activate translation of those hypertrophic markers such as collagen type 10 and MMP13. So we created a genetic vector that would hijack the actual RUNX2 pathway. And the genetic vector, it has a basal promoter, which is a COL10 basal promoter, which I said is a quintessential marker of chondrocyte hypertrophy. And it's also been well characterized in its role in the onset of osteoarthritis. Upstream of the basal promoter, we use a number of transcription factor binding motifs, or TBMs, for RUNX2. And then we use this engineered promoter to drive the expression of a short hairpin against RUNX2, which essentially causes an intracellular feedback loop that hijacks the currently expressed RUNX2 to express the short hairpin, which will then inhibit translation and decrease the expression of these hypertrophic markers, with the hypothesis that by decreasing the expression of these hypertrophic markers, we can increase the retention of matrix that's produced and give us a more robust cartilage tissue engineered tissue. And so first we wanted to show that the gene circuit was specific for chondrogenesis. And so we put our engineered promoter upstream of the green fluorescent protein and induced chondrogenesis in mouse ATDC5 cells, and demonstrated that we only see expression of the GFP where we see chondrogenesis occurring within these cells, and it only occurs after the onset of RUNX2 expression that we've verified previously. We also show that there's absolutely no expression of GFP without induction of chondrogenesis, so our gene circuit is completely dormant until chondrogenesis induced and RUNX2 expression is upregulated. Next, we wanted to show that changing the sensitivity of the gene circuit can be achieved by just changing the number of transcription factor binding motifs, or these cis enhancers. Again, putting them in ATDC5 cells, we compare them to a scrambled control, which has a non-functional short hairpin. So we should see the activity of RUNX2 with our scrambled controls, and then the suppression level of RUNX2 with our actual samples. And what we found is that as we increase the number of cis enhancers, or those transcription factor binding motifs, we do indeed get increased levels of RUNX2 suppression. And this is further borne out in the level in increased suppression of our COL10 marker of hypertrophy by our high level of suppressing gene circuit. We also see that as we increase the number of cis enhancers, we have increased levels of RUNX2 protein suppression and increased levels of matrix retention using our high level of suppressing gene circuit in these ATDC5 cells. So now that we've shown that this system works in mice, we wanted to show, of course, that it works in human MSCs. And so we took our low and high level suppressing gene circuits and put them in human mesenchymal stem cells, which were cultured in a well-known spheroid or cell pellet culture for 21 days, which is a typical assay for chondrogenesis of cells. And basically what we found with these is that our gene circuit does not inhibit chondrogenesis in any way, as shown here, and that our level of suppression is similar to what we saw in mouse cells and increases with increasing number of transcription factor binding motifs. So we can control the level of suppression of these cells, which we have shown regulates the response of these cells at different RUNX2 suppression levels. But today I'll focus on just the results from the high level of suppressing gene circuit. So our next question, of course, is, does our gene circuit resist hypertrophy? And to test this, we use a well-known additive that induces hypertrophy in chondrogenic cells, which is T3, a hormone, and we give them beta-glycerol phosphate, which is a phosphate donor, so that they can mineralize their matrix, as a hypertrophic chondrocyte is known to do. So again, when we put the gene circuit in, there's no effect on normal chondrogenesis or matrix production. But after we induce hypertrophy in these cells, we see that we have an increase in matrix production with our actual functioning gene circuit shown all the way to the right, compared to the scrambled controls shown in the middle and the wild type shown to the left in white. We also see that there is a general decrease in the amount of mineral produced by these cells, as shown on the bottom right here, which means we are inhibiting their ability to undergo hypertrophy. Further looking at gene expression, we first looked at the expression of the chondrogenic markers collagen type 2 and aggrecan, and we see that collagen type 2 expression is significantly upregulated with our gene circuit. Even without hypertrophy induction, there is some loss in expression of collagen type 2 with hypertrophy induction, but it's still significantly higher than our groups that do not receive the active gene circuit. And as for aggregate, we can see also that looking at protein expression, that we do have retention of this collagen type 2 signal after 21 days. Next, we look at aggrecan expression, and there was no real effect. Hypertrophy does decrease aggrecan as we expected to see, but the gene circuit had no effect on this compared to our wild type cells. Then looking at our hypertrophic markers, looking at RUNX2 and MMP13, we saw that in response to hypertrophy in both our scrambled and our functional gene circuit, that we had an upregulation of RUNX2 gene expression. And we really think that this is a response to the fact that we are using the local RUNX2, our gene circuit is acting like as a sink, and the cell's response is to produce more RUNX2 to fill the void. But we do see that the gene circuit is preventing that this increased expression of gene expression, this increase in gene expression is not translated into protein expression. When we look at MMP expression, it's highly upregulated by our hypertrophy-inducing media, and this is significantly downregulated or abrogated by the gene circuit in terms of gene expression as well as protein expression. And so we really think that we are maintaining not only the gene expression of collagen and aggrecan, but we're also preventing the degradation of the existing collagen and aggrecan by preventing MMP expression. Next, we looked at the effect of the gene circuit in long term. So again, we used our high-level suppressing gene circuit and cultured it over 70 days. And what we found is that the gene circuit is fairly active for the first 50 days of culture and then seems to decrease after that point. But during that time, we see, of course, robust matrix production and increased pellet size with both our scrambled and our gene circuit. And we see by 70 days, even with just the scrambled controls, we have increased expression of and retention of our ECM matrix proteins. So even in long term, our gene circuit is regulating the matrix production and retention of these cells. We confirm that over this culture period, we have consistent downregulation of RUNX2 gene expression, as well as the expression of its target gene, collagen type 10. And we can see that there is significant downregulation of COL10 expression or protein expression with the gene circuit over the 70-day period. When we look at our chondrogenic genes, aggrecan and collagen type 2 are overexpressed with respect to wild type in all cases, except for day 35 with our scrambled gene circuit compared to wild type. And again, we see that our aggrecan expression is upregulated in this process, and our call to retention is higher using our gene circuit. And so just a summary for this part is that we've shown that our auto-regulated gene circuit, which controls on a cell-to-cell basis, the hypertrophy of these cells is actually functional and supporting our hypothesis, which is that we can increase the amount of matrix that these cells produce and retain. And it's likely that we are inhibiting the degradation of that matrix by inhibiting the expression of MMP13 protein. And so we've come up with a way that the cells can respond to their matrix or their environment on an individual basis and produce a more robust tissue. But then we asked the second challenge, which is we're going to take this engineered cartilage or these cells that we've engineered, we're going to inject them into a joint or repair tissue into a joint that's already inflamed. And not only is it inflamed, but it's going to stay inflamed. So unfortunately, once joint injury occurs in articular cartilage, we have the release of signals known as danger-associated molecular patterns, or DAMPs, that activate macrophages and fibroblasts within the synovium, causing them to differentiate into inflammatory cells. Chondrocytes respond to these cells by degrading their matrix through upregulated MMP13 expression, for example, and also express their own inflammatory factors, which will cause the release of more DAMPs, which will activate more cells. And so we have a vicious cycle of inflammation going on. And so what we know about inflammation during osteoarthritis, if we look just down at the cellular level, is that it's regulated also by RUNX2. So we know that TNF alpha and IL-1 beta through the NF-kappa B pathway will activate RUNX2, giving us an inflamed chondrocyte that will degrade its ECM. So we really wanted to understand whether or not our gene circuit could help our cells resist the impact of inflammation. And to do that, again, we used our same system, and we treated either with IL-1 beta or TNF alpha for 72 hours after 21 days. And with wild type, we see that there's a general decrease in matrix retention with IL-1 beta treatment, but not with TNF alpha treatment. And when we give them the gene circuit, we see again this upregulation of matrix expression in our with our gene circuit, even in our control cells. And in response to IL-1 beta, there is a significantly upregulated expression of or retention of matrix proteins, but it's not very high. So we don't reach wild type levels. But we do show that when we look at protein expression, that we have, again, our decrease in RUNX2 with treatment with IL-1 beta. And we see that there's no expression of MMP or lowered expression of MMP13 in the gene circuits in response to IL-1 beta treatment. But our best and most exciting data, of course, comes from the fact that we have this high level of aggrecan retention, even in response to inflammation. So basically what we're seeing now is that we can also use the gene circuit to not only resist the impact of hypertrophic signaling, but also this inflammatory signaling. And so this brought us to our next area of interest, which was our cell systems and understanding crosstalk amongst cell systems. So it's becoming more and more important that we understand and we study the joint as an organ. We know that macrophages will influence chondrocytes, and conversely, chondrocytes will influence macrophages, so on and so forth. And all of these systems have been characterized in sort of in a vacuum and not really with the other systems. So we really wanted to understand if we're reprogramming our stem cells and injecting them into the joint, what effect will it have on the inflammatory macrophages? And so that's the focus of this next part of the talk, is understanding this crosstalk between these two cell types. And the way that we accomplished that was in a parallel in vitro model. So we took THP1 monocytes, we induced an inflammatory phenotype of them, also known as an M1 phenotype, which we confirmed by the downregulation of CD68 and upregulation of iNOS, as well as IL-6 gene expression. And in parallel, we took HMSCs and reprogrammed them with our high level of suppression gene circuit, cultured them for 21 days. After induction of inflammatory or the inflammatory phenotype within our THP1 cells, we took their conditioned media and we put it on our 21-day chondrocytes for 72 hours, and we looked at what effect it had on matrix retention. And we were really excited to see that we had almost a complete abrogation of loss of matrix with the gene circuit under these conditions. And the reason that we chose these conditions because they're more similar to what we see in the joint, rather than treating them with something like IL-1 beta or IL-1 alpha, which had little effect on matrix at these supraphysiologic conditions. And more importantly, we see that it's not just IL-1 beta or IL-1 alpha mediating this loss in matrix. There has to be other components in this cocktail that we're giving them that are either working individually or synergistically to not only degrade the matrix, but activate the gene circuit. When we look at the expression of inflammatory cytokines by our chondrocytes, we see that we have downregulation of MMP13, as we've seen previously, but also the inflammatory markers IL-6 and NF-kappa B. So these are significantly upregulated by our conditioned media, and this response is abrogated by our gene circuit, which was really exciting for us. And of course, I did say crosstalk, so we want to understand what effect these activated chondrocytes now have on the inflammatory environment in the joint. And so we took double conditioned media from these after 72 hours of treatment and put it on a new set of inflammatory macrophages and looked at the expression of pro and anti-inflammatory markers. And we were really excited to find that across the top, the pro-inflammatory marker iNOS decreased with our gene circuit, and across the bottom, the anti-inflammatory marker CD206 increased. And so it looked like we have a repolarization of the macrophages in response to the secretome from our activated reprogram HMSCs. And we wanted to confirm this, so we performed RNA-seq on the macrophages. And we did see general downregulation in the inflammatory response and an increase in cellular biosynthesis. When we look more closely at the genes that are being expressed, we see that we have inflammatory markers that are upregulated when in our M1 conditioned media, shown in gray. In white, we see downregulation of some of these inflammatory markers, but more importantly, we see that with our gene circuit, we have even further downregulation of these inflammatory markers. And with either the wild-type conditioned media or our gene circuit, we have upregulation of anti-inflammatory markers. And so basically, in summary, what we've shown now is that we have that autoregulated RUNX2 suppression can not only inhibit the effects of hypertrophy stimulating exogenous cues, but it can also abrogate the effects of inflammatory stimulus. And more importantly, what we see is that we have this repolarization of macrophages, which can then be used to increase the anti-inflammatory phenotype and hopefully create a healing environment rather than an inflammatory environment, and decreasing the onset of PTOA. And really, that's where we're going next with this. So the clinical relevance we really need disease-modifying effects for PTOA. So not just being able to repair the cartilage, but can we actually use these cells to create a less inflammatory environment? And so we're currently looking at cartilage defect repair with these cells. That's ongoing. And we also want to understand whether or not we can prevent the onset of PTOA, possibly with an ACL rupture model, with these cells after we repair a cartilage injury. And with the ACL rupture model, we can also ask the question, can we repair cartilage in this inflamed environment? So can we repair cartilage in an already activated osteoarthritic joint and still get functional cartilage healing? And so currently what we're doing is injecting the cells directly into the joint. And looking at whether we can either prevent or reverse joint-wide disease after OA stimulation. And I'm doing this with Tristan Metz here at University of Michigan. So in the vein of looking at whether or not we can repair cartilage in aged or inflammatory joints, we started to look at whether or not we can use the gene circuit to improve the cartilage producing ability of aged chondrocytes. And so essentially we've been taking donor tissue from patients that are getting their knee replaced, isolating the cartilage from their relatively healthy regions and looking at their ability to produce matrix. And what we can see is that compared to the beautiful histology that I've been showing you, we don't get robust matrix production and we get a more fibroblastic phenotype. And so we really asked the question, what happens when we regulate RUNX2 in these cells? Can we use that to reverse this aged phenotype of matrix production? And so we looked at first putting our low level of suppressing cells, and we do see some restoration of the ability of these cells to produce matrix. And when we use our high level suppressing cell, we almost completely recover the ability of these cells to produce a robust matrix. And that's borne out here in looking at their matrix production over this large period of time. We also looked at again, the expression of the cartilage-specific molecules SOX9, COL2, and aggrecan, and showed that they were all significantly upregulated with our gene circuit compared to wild type, and that RUNX2, COL10, and MMP13, our hypertrophy markers, are significantly downregulated. So the gene circuit can be used to understand or excuse me, to give these cells the ability to produce a robust ECM and hopefully increase the chances that we can repair aged chondrocyte in these patients or aged cartilage in these patients. And so last thing I'm going to talk about is how we're moving forward with the gene circuit and how we're moving forward with, redesigning the gene circuit for a more sophisticated regulation of cell function. And the way we're doing this is to use data-driven models to understand how cells transition between the different cell states, and then use that to redesign our gene circuit. And one of the things that's very important to remember is that the microenvironment has a huge effect on cell response. So everything we've done has been in a scaffold-free environment. But we know that there are very many factors that integrate to regulate the final chondrocyte phenotype, and understanding how those factors regulate this process is the next phase of these studies. And to do that, we're using a biosynthetic prechondrogenic stem cell niche device for my lab. And we devised this so that it replicates the actual developmental environment of stem cells undergoing chondrogenesis, which we would find in the limb bud or in the temporal mandibular joint in the jaw. And through some really elegant studies that have gone on in the past, we know that this environment is predominantly collagen type 1, fibronectin, and hyaluronic acid, and that these factors together mediate condensation and chondrogenesis. And so to incorporate that into an environment, we took 8-arm PEG. We used a really simple PEG system that was acrylated. We used sialylated hyaluronic acid and methacrylated collagen type 1 in an effort to allow those two to interact with each other as we would see in a normal cartilage environment rather than just have them freely attached to the PEG hydrogel. And so through microtype additions, we get interactions between the collagen and the hyaluronic acid, as well as the collagen or the hyaluronic acid and the PEG. And then afterward, we incorporate the cells, UV them so that we can come up with a solid hydrogel. So this is a simple system in which it's easy to incorporate stem cells. We've shown that with this system, we can change the stiffness by changing the percent PEG, and we can also change the composition, and we can change these independent of each other. So we can really understand the contribution of collagen type 1 and stiffness and aggrecan and stiffness on this process. We've shown that this complex changes in the microenvironment have complex effects on the expression of cell surface receptors, condensation markers, so specifically NCAD and NCAM, which are very important for the condensation process, and finally chondrogenesis. And so this system is very versatile in allowing us to study the impact of these environmental factors on stem cell chondrogenesis. Since stem cells condense before they undergo chondrogenesis, we further added sophistication to the system by patterning the cells within the hydrogels using ultrasound, which is something we can talk about offline if you're interested. Culturing these and then understanding how these individual parameters, in addition to the pellet size, cell seeding density, and other factors, will affect how these cells differentiate. Once the cells are in this system, we're using a complex set of analyses really to understand how the microenvironmental parameters that we investigated, which were matrix stiffness and loading, as well as patterning, affect the differentiation of these cells. And really the reason for doing is to understand how the cells differentiate between these different cell states so that we can regulate the cell states genetically. So we would be, our goal is to look at the secretome, this transcriptome of both mRNA and microRNA, so we understand both transcription and translational regulators, as well as the stiffness of the matrix over time. With that, in collaboration with computational medicine here at Michigan, we're using a data-guided algorithm that will allow us to predict which transcription factors permit the cells to transition from cell state K to cell state K+1 and back from and back to K if necessary, so that we can then design the gene circuit to target these specific transcription factors. So basically our outcomes would be transcription factors and microRNAs that modulate cell state transitions. We'll be able to calculate those, as well as the time course of the expression of these transcription factors. And that time course is really important for developing or developing the promoters of these gene circuits. If we can understand the time course at which transcription factors are being regulated, we can design the promoter to have a combination of transcription factor binding motifs that targets it to a specific cell state. Our transcription factors and microRNAs can then be used as effectors that we either overexpress or suppress in order to regulate cell phenotype. So this is the direction that we're moving the gene circuit technology to, so that we have a more elegant and sophisticated system in which we can tightly regulate the phenotype of these cells. So I would like to thank the members of my lab who were mental in developing these techniques, especially Beaming Wu and Gertrude Core. My list of funding sources, and I would like to thank you and ask you if you have any questions.
Thank you very much. Actually, that was very impressive. I really liked it. Really enjoyed that synthetic gene circuit that you had ongoing there. I think
Great.
a lot of potential there for discovery. Let's see, does anybody have any questions? Please feel free to open up or unmute and ask. I could definitely start off though. So when you use the IL-1 beta in your initial circuit in your inflammatory milieu, I mean, a lot of people use IL-1 beta to look at the effect of matrix protein degradation and so on. And I think most of us tend to use supraphysiological levels in order to induce some sort of response. I'm not sure what levels you used in your experiments, but you know, maybe the reason why you didn't see any major you saw changes, but maybe the changes weren't as profound because of the experimental conditions, the amount you're using. In OA joints, I know it's like less than a nanogram per ml in the synovial fluid. At least that's what has been shown.
Agreed.
That could be one explanation. I'm not sure, but you did. I like the way you followed up by using the macrophages, the conditioned media, to show that. That was pretty. It's more of a comment. That was pretty interesting. The question I had though was actually based on the other transcription factors. So for the in hyperchondrogenesis, or so you had in hypertrophy of the chondrocytes, you did show a nice schematic summarizing some of the transcription factors that are upregulated in addition to RUNX2. So I was wondering if you had a chance to look at others such as FOS, if you use that in your circuit to see if that had any effect.
No, we planned on doing FOXA2 and MMP. No, it was FOXA2 and something else. I can't remember. Which are upstream of RUNX2 because, of course, once RUNX2 is expressed, we're starting to get hypertrophy. But we never were able to successfully develop those gene circuits. So we haven't. And that's the other reason for the more sophisticated analyses with the data-guided algorithm so that we're not just shooting in the dark, right? So basically, we know a lot of things based on fundamental studies that have been going on with the growth plate for decades, but we don't know a lot. We still don't know a lot of things, and we don't know all of the differences that are upcoming with chondrogenesis, especially with donor variability and things like that. And so this is a way to give us an omics approach to give us as much information as possible, and then a list of transcription factors that we should try in the gene circuit. So a more intelligent, systematic way of approaching it, rather than using the knowledge that we currently have. We're using data sets that already exist, plus our own data sets.
No, it's brilliant. It is quite impressive. The other question was for the RUNX2. So RUNX2 in articular cartilage, there is somewhat of a gradient from the superficial layer to the calcified layer. So I was wondering if you foresee any issues when you're doing your, tissue engineering or injection of your MSCs. Do you foresee any issue with, let's say, the calcification of in the calcified layer of the cartilage, and whether that may impact the biomechanics of the tissue?
That's actually a really good question. We hadn't thought about that. We haven't tried making an osteochondral tissue yet. What we could do, because we can regulate the level of suppression of RUNX2, we could put high level of suppression in the top, at the bottom, low level of suppression, and then no suppression underneath that. And that could be a way to make an osteochondral tissue. But that's actually a really good question that we hadn't considered before. So thank you.
No problem. Thank you.
Mike,
Anybody
we have
else?
a question. We have a question in the chat from Fatima.
Yeah.
So were the human MSCs derived from a single donor or multiple donors? And if multiple, did you observe any variability in ranks 2 outcomes across donors?
So there are multiple donors. That's a good question. We haven't noticed any significant differences in rungs 2 levels. And the response to the gene circuit is essentially the same compared to its internal control, right? So we would see a 60% loss of matrix, whether they produced an enormous amount of matrix or a small amount of matrix, it was still 60% loss. So yes, there is going to be some differences in the amount of rungs 2 that's expressed and therefore how much the gene circuit gets activated. And we also have to consider the heterogeneity of the system. So everything we're doing right now is bulk, it's not single cell. And the heterogeneity in that system might also be contributing to differences in rungs 2 that we would see. So single cell RNA-seq would give us a better idea in answer to that question. And we're doing that now for aged articular chondrocytes specifically so we can understand that and design gene circuits that are more sophisticated for those cells.
So if there are no more questions, we thank again our speaker for coming to present for us. Thank you very much. It was very interesting. So if you stay with me a bit online for a few minutes, I have some late announcements. As I mentioned at the beginning of the seminar, this is the last for this year. And of course, we have some awards for our students that presented. So I'm gonna show you the awards. So for the presentation, we have a first prize award for $200 for Najad. Najad is a PhD candidate in Dr. Harley's lab, and she had a score of 4.56 based on your votes. We have 2 second prizes of $175 for Alexandra Dimmer in Dr. Poliganda and Dr. Poenaro's labs with a score of 4.53. Matthew Zakaria in Dr. Edwards Harvey lab and Dr. Merle lab with a score exactly like Alexandra's, 4.53. And we have a third prize for $150 for Georgi in Dr. Hammerling's lab with a 4.49. Additionally, we have special mentions of $75. Tanais, Cassandra, Anthony, and Zara, thank you for presenting with us. Of course, we have our seminar question award for, Fatima and Mashid. They're gonna get $50 each. We have an additional award for Students' Council who helped us to organize the seminar for their involvement. Muskan, the president, Pega, the VP academic, Eliane, VP external, and Temitope for health, wellness, equity, and diversity representative. This will be 100 awards. They also contribute at other events like Research Day and Opening Night. And since we're at the Research Day, please don't forget to register for the Research Day. We will have out the results for the speakers and the presentation probably in the next few days. We will send out the emails. What I would like to ask you, if you can't attend, once you receive the email and you'll be invited to present, if you can't attend, let us know, because we will replace with somebody else. If you come in the last minute, that place will be not covered. Well, I wish you nice summer. We'll see each other in the fall with the next seminar, and also good luck with your exams.
And thank you again, Dr. Coleman, for accepting our invitation and for that wonderful presentation.
Thank you all. Have a good summer.
Bye.
Hey, you too. Bye.